All right. Hello, everyone. Thanks for joining us today for the Cellular Respiration episode of the Makella podcast. This will be an interesting episode today, as due to the pandemic, we are recording over a Zoom call.
Mackenzie, I have never heard of cellular respiration. This should be an interesting and informative episode for me and our listeners.
Oh, Ella, how do you not know what cellular respiration is? If I remember right, the equation for cellular respiration starts out as sugar and oxygen as reactants, and then it gets converted into carbon dioxide, water, and ATP as products via a chemical reaction. It's important to remember, however, that the purpose of the reaction is not to make CO2 and water, but the production of ATP itself.
Oh yeah, that rings a bell. I remember learning about that in AP Biology.
Okay, so do you remember the first step then?
No, I don't.
Okay, so basically we start out with glucose and oxygen that our body takes in through eating food such as sugars and carbohydrates and breathing in the oxygen.
Okay, so what's the point of cellular respiration?
So our bodies need ATP, adenosine triphosphate, which is a form of energy. The ATP powers many of the functions at a cellular level all throughout our body. These functions allow our body to do the things it needs to do. The only thing I don't really know is what happens after this.
Oh, that makes sense. And I think I remember this part. The big cellular respiration cycle is made out of smaller steps. The first step is the 10 steps of glycolysis, which happens in the cytosol or the cytoplasm.
Ooh, what are those steps?
To start the 10 steps of glycolysis, we need a little bit of ATP, about 2, that have already been made from previous cycles. This ATP breaks apart the glucose in the equation.
Wait, why does the glucose break apart?
We need the glucose to break apart into 2 3-carbon sugars, so we can gather the electrons, which we will save for later.
What gathers the electrons, and why do we need them for later?
The breaking down of glucose happens in the first 5 steps of glycolysis. The glucose is broken down into 2 3-carbon pieces made from the molecule. So there are coenzymes, which are like these little school buses called NADH and FADH. In step 6 of glycolysis, they carried the electrons that were taken out of the breaking down of glucose. After these broken up carbon glucose halves produce a total of 4 ATPs.
Okay, that makes sense. But after the steps of glycolysis, what happens to that glucose?
I was just about to tell you that. The broken down glucose is called pyruvate, which is made out of 3 carbons.
All right, I get it. And if you don't mind, can I talk a bit about the second step?
Yeah, that would be great.
So, the next step is oxidation, or losing electrons. This all starts in the mitochondria.
Wait, isn't that the powerhouse of the cell?
Yep, you got it. So the CO2 groups that are part of the glucose get detached in the mitochondria. Once the pyruvate gets oxidized in the mitochondria, again, meaning it loses electrons, it turns into acetyl-CoA. Each pyruvate molecule loses a carbon in the form of CO2. When this enters the mitochondria, the acetyl-CoA gets broken down and carbons leave, like I said, in the form of CO2. Once it enters the mitochondria, the same school bus from before in glycolysis, NADH takes away more electrons.
Wait, doesn't that take a super long time?
Yeah, it actually does. But there are certain enzymes that help make this process faster. In fact, a super important enzyme in this process is SCOA. It attaches to the molecule and helps it move along to the next stage.
Now let's get into the citric acid cycle.
Ooh, exciting.
Yes, this part is very exciting. So once the pyruvate gets converted into acetyl-CoA in the mitochondrion, the acetyl enters the citric acid cycle, also known as the Krebs cycle.
Oh, yeah, that's right. Doesn't the citric acid cycle run through twice for each glucose precursor?
Yes, there are 8 reactions in each cycle that are held in the mitochondrial matrix. In step 1, the acetyl-CoA gets converted into citrate, a weak organic acid. After that, in steps 3, 4, and 8, NADH, and in step 6, FADH2, and both of those are coenzymes, get released into the electron transport chain.
Oh, that's really interesting. So, are there any byproducts of the citric acid cycle?
Yes, there are. In each turn of the cycle, there are 2 carbons found in citrate that get converted into CO2. If you remember the original cellular respiration equation, you'll see that there is carbon dioxide as an excess product of the chemical reaction.
Okay, now let's talk about where all of those electrons went once they are carried by the school buses or coenzymes.
Yay, I was wondering where they all go. I remember that there is one last step in aerobic cellular respiration. Isn't it oxidative phosphorylation?
Yes, in oxidative phosphorylation, the electron transport chains accept all of the electrons from NADH and FADH. This part of the process forms about 90% of the ATP.
I know this part of the cycle is produced by redox reactions that include oxidation, which is the loss of electrons, and reduction, which is the addition of electrons to another substance. Do you think you could go a bit more in depth in it?
Yeah, this part is a little bit complicated. So the school buses, NADH and FADH, bring all of the electrons into proteins that are in the inner membrane of the mitochondria. In the membrane, there are integral proteins packed along the membrane in a formation called the electron transport chain. And this chain, the next protein, is more electronegative than the last one. This electronegativity allows the electrons to move from one protein to another. This motion is called the proton motive force. The proton motive force moves the hydrogen ions from from a low to a high concentration into the outside of the membrane. Each time the electrons flow from one protein to another, a hydrogen is pushed against the natural gradient and into the outer membrane space. This builds a high concentration of hydrogens in the outer membrane space.
Oh, cool. I have a question though. How do hydrogens get back into the membrane? I know that they probably don't want to pass through the mitochondrial membrane since they don't like the hydrophobic tails surrounding it.
Great question, Ella. You are totally right. So this is where one of the most important parts of cellular respiration comes in. The hydrogen passes through the phospholipid bilayer, made out of those hydrophobic tails you were talking about, in something called an ATP synthase.
Oh, what's that?
So ATP synthase is a specialized channel that lets hydrogen ions through the membrane. The process of these ions going through the ATP synthase is called chemosis.
But I'm confused. How does this process help generate ATP?
So the ATP synthase channel has a rotor that spins, kind of like a boat. When a hydrogen goes down the rotor, it causes it to spin. The motion of this spinning attaches an inorganic phosphate to ADP, adenosine diphosphate. The combination of this ADP and the phosphate is ATP.
Oh, wow, that is totally amazing. How much ATP in this process is actually made?
Well, this part's kind of like a math equation. As we talked about before, during the steps of glycolysis, a total of 2 ATPs are made, since it takes 2 ATP to start it, and 4 ATP are made. And on top of that, the citric acid cycle produces 2 ATP. And ATP synthase channel, like we just talked about, produces around 32 to 34 ATP, which leads us to a total ATP an organism makes somewhere between 36 and 38 ATP.
Wow, that is a lot.
Okay, but if I remember right, there are 2 types of respiration?
Yes, you are totally right. There is aerobic respiration and anaerobic respiration. Aerobic means that oxygen is consumed, but what happens if there's no oxygen present?
Would the organism like archaea and some bacteria use anaerobic respiration or maybe fermentation?
You got it right on the mark. Fermentation is an extension of glycolysis that allows a continuous generation of ATP by the substrate level phosphorylation of glycolysis. In this process, it can regenerate AD+ by transferring electrons from NADH to the pyruvate.
Aren't there two types of fermentation, alcohol fermentation and lactic acid fermentation?
Yes, alcohol fermentation happens when the pyruvate is converted into ethanol. Without oxygen, CO2 is released from the pyruvate, then converted into acetaldehyde. The acetaldehyde is then reduced by NADH into ethanol. Eventually, there is such a buildup of ethanol that everything gets killed and turns into alcohol that people drink.
Oh, yum. So what happens in lactic acid fermentation?
In lactic acid fermentation, the pyruvate is reduced directly by NADH to form lactate.
Wait, but why do we have to form lactase? Why don't we just stop at pyruvate?
Oh, great question. Us humans don't just stop with pyruvate because they make, wait, can you? Oh, great question. Us humans don't just stop with pyruvate because when we make lactate, the electrons from NADH are able to be moved into lactic acid. Then the NAD plus is free to go pick up more electrons so we can do it all over again. This is the same case with alcohol fermentation.
Oh, from running, I've heard some people mention lactic acid. How does lactic acid fermentation relate to our bodies?
The lactic product is then mostly oxidized by red muscle cells, with the remainder exported to the liver or kidney cells for glucose formation.
Oh, okay. Well, I think that's it. This has been such a great time, and I'm so glad to have learned about cellular respiration with you. Hopefully,
Thanks,
it gave our viewers a better understanding.
thanks me too. Next week, I'm very excited because we will be learning about the nervous system.
Hooray!